Microscopy 1. Introduction to Microscopy. Microscopes play a crucial role in biology by allowing scientists to observe structures that are too small to be seen with the naked eye. Over time, microscope technology has advanced significantly, leading to improved magnification and resolution. 2. How microscopes work. Microscopes help in viewing and studying small objects by magnifying them. Here are some key points about how microscopes function. Light microscopes use light to visualize specimens, making them useful for examining cells and simple structures. Electron microscopes provide much higher magnification and resolution than light microscopes, enabling the observation of intricate subcellular structures like the nucleus, ribosomes, and plasmids. Advancements in microscopy. The development of electron microscopes has allowed for a more detailed understanding of cellular structures. 3. Magnification and standard form in microscopy. The magnification of an image is calculated using the formula magnification equals image size real size. Example, if a cell has a real width of 0.02 mm and its image width is 8 mm, the magnification would be 8 mm 0.02 mm equals 400 frac. Writing numbers in standard form. When working with very small or large numbers, standard form simplifies representation. For instance, 0.0025 can be written as 2.5 times 10⁻32.4. Preparing a microscope slide. Before observing a specimen under a microscope, the slide must be prepared correctly. Place a small drop of water in the center of the slide. Peel a thin epidermal layer from an onion using tweezers. Position the layer onto the water droplet. Add a drop of iodine solution to enhance visibility. Carefully place a cover slip on top, avoiding air bubbles. 5. Using a light microscope. To observe a specimen under a light microscope, follow these steps. Secure the slide onto the microscope stage. Select the objective lens with the lowest magnification. Use the coarse adjustment knob to bring the slide into focus. Look through the eyepiece and fine-tune the focus using the fine adjustment knob. If necessary, switch to a higher magnification lens for a more detailed view. 6. Drawing microscopic observations accurately. When recording microscope observations, follow these guidelines. Use a sharp pencil for neat drawings. Outline structures with smooth lines, avoiding unnecessary shading. Clearly label key features like the nucleus and chloroplasts. Maintain proportion and clarity in drawings. Include a title and a scale for reference. Record the magnification used. 7. Importance of light microscopes. Light microscopes provide a valuable tool for biological research, offering a balance between accessibility and detailed visualization. Preparing samples correctly ensures accurate observations.